Hello everybody, welcome back. Today we're going to talk about my favorite subject, genetics. Genetics is this huge field, so many applications. We are going to just kind of scratch the surface. Traditionally, genetics can be quite challenging for a lot because once again, we're not talking about cells, but we're talking about things that are inside the cells. And so the challenge here is trying to find like this moving picture of the concepts that we are going to talk about. We are going to talk about just the basics, just enough for us to understand some of the things that have application outside of hardcore molecular genetics, right? But again, try to find, rather than looking at words, try to find things that move, right? And try to imagine what's going on with respect to DNA and the various applications of it. Okay, so without further ado, let's get going. All right, like with anything, in order for us to understand the subject, we need to talk about some of the definitions, right? So like what is genetics, all that kind of stuff, right? So genetics is the science of heredity, right? Basically, what are genes, how they carry information, how this is copied, how's it passed, how does it change from generation to generation. When we're talking about DNA, we're talking about the genome. All the genetic information within a cell, which includes things like chromosomes and plasmids, right? We will define some of these a little bit later. And ultimately, we talk about genes, which are segments of DNA that carry a functional products. And for us, functional products typically are, where's my cursor? There you are. Proteins, right? That's for us important, right? Because remember what proteins are. Proteins are our cell tools. Almost nothing happens in a cell without a protein, right? Or RNA molecules, right, which are also information that's carried within DNA. Now, ultimately, when we say a product is made from a gene, we say that this gene is expressed, right? And the gene expression is very important, of course, because we need to make sure, or we need to get and use the protein products or the RNA products from the genes themselves, right? But gene expression is highly dependent on the environment, right? So here's a really good example, something that you can apply in your everyday life or talk to your kids about. In our mouth, we have a bacterium called Streptococcus mutans, right? It's there. You absolutely cannot get rid of it, so don't even bother, right? In the lab, we can, in your mouth, it just eats the sugars of basically all the food that you eat, right? One of the very important sugars for it is this thing called sucrose. Sucrose is table sugar. It's that sweet sugar that we find in candy, right? The stuff that tastes so good, right? And in the lab, we can show the importance of sucrose because when we grow it here on glucose, that's what our colony looks like. This little thing here, that's our Streptococcus mutans, right? Billions of cells sitting on top of each other. But when we grow it on sucrose, look at what happens to our Streptococcus mutans. Same cells, same bacteria, right? But this time what happens is that it's able to form this sticky layer, right? This extra polycellular saccharide that it can surround itself with, right? And this is plaque. So what we're looking at here is plaque on a plate. And it uses this plaque to stick to our teeth. And then leave it long enough, it'll turn into tartar, which then turn into damage to your teeth or tooth enamel that we call cavities, right? So this is proof literally that sugar is bad for your teeth, right? So sugar is really bad for your teeth, right? However, understand this here. The genetics of both of these cells are exactly the same, right? Nothing has happened to this cell that has changed what it is, right? All that has happened is the environment that it finds itself in. Glucose versus sucrose, right? We've just changed the sugar that we're feeding it. And then now that has changed the expression of the genes to lead it to change its way to the way that it looks, okay? And so we have terms for this, genotypes and phenotypes, right? So genotypes are simply the collection of genes within a cell, the information that's found in its DNA. And we call this the potential characteristics, right? So going back to this, the genotype of both of these two cells are exactly the same cells. Nothing has changed, right? Phenotype is the expressed properties. Remember, taking genes and turning it into some sort of product, right? And then this expression then leads to various abilities, various ways that it looks. You know, we see it often as brown eyes, blue eyes, curly hair, straight hair, you know, all that kind of stuff. If you're lactose intolerant, all these things are different phenotypes that we see here, right? So in this case now, this environment that Streptococcus mutans finds itself in, right, has led to a change in the genes that are expressed, which then change in its phenotype or the way that it looks, right? And so in many ways we talk about genotype being the collection of DNA in the cell, phenotype being the collection of proteins, right? And the expression of the proteins is very much dependent on its environment. When we talk about DNA, right, the organization of DNA, we organize our DNA into these large structures we call chromosomes, right? We all have them, but the way they're organized are slightly differently. And again, it's way more complex than this, right? But we're going to talk about the kind of like the big surface differences between eukaryotic and prokaryotic chromosomes and how they are organized. Many, many similarities, and then some major differences, which really are not going to look like major, just sort of differences. Eukaryotic cells typically have many linear chromosomes, right? So they'll have these kind of strings of chromosomes. And then typically they're diploid, which means that, there we go, we'll have 2 copies of the same chromosome, right? So that whole 23andMe thing, you know, we have 23 chromosome pairs, right? 46 in total, right? So actually for each of our 23 chromosomes that we have copies of 2 copies of each one, right? Which is kind of useful because if, let's say, we happen to have a piece of DNA here with a really bad gene that doesn't work, right? Well, we have our second gene right over there to help compensate for that, right? So, you know, there are advantages to being diploid. And then they are packed and coiled using these things called topoisomerases, using proteins called histones. Okay, and we'll talk about that in just a second. Prokaryotic cells typically have 1 circular chromosome, so the chromosome looks like this, right? And then no nucleus. Oh yeah, shoot, I forgot. So remember the definition of a eukaryotic cell, the DNA is trapped within a nucleus, whereas a prokaryotic cell, no nucleus. Instead, it has a structure called a nucleoid, right? And then typically, prokaryotic cells are haploid, only 1 copy of the chromosome. They are also packed and coiled by, you know, quote-unquote topoisomerases using histone-like proteins. Not exactly the same, but sort of the same. So, you know, technically, for all intents and purposes, the same, but different enough that we have to mention it, okay? So then when we take these, you know, talk a little bit about packaging, about these enormous molecules. Remember, these molecules are huge. They're like 2 meters long, 1 to 2 meters. That's 3 to 6 feet of a chromosome, right, in each single cell. Right? So how do you fit like 3 to 6 feet of DNA in a single cell? And every cell has that amount, right? And so it turns out that we do this by packaging and packaging, right? This is how we're used to looking at DNA, right? This double helix, right? But this is our long, unwound version of DNA. Right? So we can take this and then we can wrap it around these things called nucleosomes. And then we can take these nucleosomes and then wrap them around things called histones. And then we can take these histones and wrap them around these things. And then we can take those and then wrap them around something else, right? And then ultimately packing it super duper tight, coiling it on each other to form a chromosome. So a chromosome is this really intense, packaged, wrapped DNA, okay? And so that's how you can take 2 meters, 6 feet of DNA and squish it into a teeny tiny cell, okay? Now there's a problem with this, of course, right? Because ultimately when we use the DNA, we actually need it to look like this because I need to get access to the nucleotides that are present in our DNA molecule, right? So when it's super duper wrapped like this, not gonna happen, right? So think of this as like one of those hoarder garages, right? You know, you can stuff so many things in your garage if you got one of those hoarder garages, boxes in boxes, which are on top of boxes, which are on top of other boxes, all shoved into the garage, right? But you want like that thing right there, how are you gonna get it? You gotta unwrap everything. You gotta move all this stuff out. I had a neighbor who was like that, by the way, and sometimes we would see all their stuff on a lawn because they need to get it a shirt that they had somewhere in their hoarder garage, right? Which then leads us to this thing called epigenetics, right? I don't want to say too much about epigenetics because it's extremely complex, but it's something that it's actually becoming somewhat common to talk about, because one important aspect of control of expression is this idea of changing of packaging, right? So sometimes it's not as simple as just deciding to turn a gene on. Sometimes what we can do is we can keep genes off by simply covering them up, hiding them in these packages, shoving them in the back of your hoarder garage, right? And we can give access by unpacking, right? So that's what epigenetics refers to, control by changing in how we package our DNA, okay? I don't want to say much more than that. Again, it's extremely complex, but in some ways they say it's very powerful in how our genes are expressed. And again, really taking the DNA that you have and making who you are. Sometimes we have this wonderful gene in our DNA, but due to this darn epigenetics, it's just not being expressed. We just haven't figured out a way to uncover it. The other part I want to list a little bit about, right? Make you aware of it, is this idea of non-coding DNA and then extra chromosomal DNA. Oh, shoot. But I'm sure you know what that is, right? There we go. Non-coding DNA, or regions of DNA that don't code for a product, right? We often used to call this junk DNA because basically we had no idea, right? In fact, one of the biggest surprises when we first sequenced the human genome, we thought, okay, humans, we are super complex, extremely powerful creatures. We have conquered the world. Our enormous genome must house literally millions and millions of genes. So it turns out it's only like 20,000. And it was a huge surprise. Like 98% of our genome is non-coding, right? Turns out that this junk DNA is not really junk, right? It's very important in things like regulation. And so that whole epigenetics thing I was talking about, super important for that. We don't really understand it though, right? So sometimes we still call it junk DNA, right? And so in fact, our knowledge of DNA is so minimal, right? We know so little of how the things in our DNA lead us to who we are, right? So just be wary, by the way. There's a lot of DNA companies out there that'll say, oh, we'll sequence your DNA and tell you what diet to eat, right? You know what they're doing? They're kind of guessing based on what they know, and it's kind of limited, right? Prokaryotes, much more efficient, right? So they have way less junk DNA, right? Their genomes tend to be much smaller. They don't really have a lot of space to waste. Epigenetics is not a big thing for prokaryotes. So for them, the non-coding regions are much, much, much smaller, right? And then finally, extra chromosomal elements. These are DNA molecules that are not in the main chromosome. So we've seen these things as mitochondrial DNA, chloroplast DNA. These are elements of DNA that aren't in our chromosome but are still a very important part of a genome. Little ambiguous. Sometimes when we say genome, we refer to as well as mitochondrial DNA. Sometimes we don't. And why we have these weird distinctions, I don't know. And then finally, sometimes viruses are part of our genome, and plasmids as well. And we'll talk about plasmids a little bit later. So let's get to the good stuff. How do we use DNA, right? Well, the DNA has a couple major functions. One is it's the genetic material responsible for inheritance, right? So, we need to pass on this DNA, so it needs to be copied properly before being passed on, right? Because each cell needs to have the information. Each cell needs to have DNA. But it also carries information to make products, right? And these products, again, typically are protein. That's the most important one for us, but also RNA. And that's very necessary for cell growth. So each cell needs to have this information, right? But then, which then means, of course, we need to have the information, but we also need to copy this information before we divide the cell, okay? So this then leads us to our one kind of I guess, our unifying theory of biology, what we call the central dogma of biology. You know, biology has such few rules. You know, the thing I love about biology is there are always exceptions to the rule. Always, right? So, you know, we talk about how like each cell, you know, life cannot exist above boiling. Well, it sort of can, right? We talk about how everything has DNA as its genome. Well, there are actually some viruses that don't, right? And, you know, we talk about how like we need the sun to make carbohydrates. Well, there's a group of bacteria that actually don't use the sun at all to make carbohydrates, right? So there's always these rules, these exceptions, these rule breakers. So biology, ultimate rebels, because life finds a way, right? But we say, this is the one unifying rule, central dogma. DNA is used to make RNA, which is used to make protein. Sometimes because we want to be all like cool about it, we say DNA to RNA to protein, and that's like our central dogma. And then we did learn that viruses have RNA genomes, as we learned about, right? But technically, the RNA genomes of viruses need to be converted to DNA first before we do this and this. So we can say, aha! Even for RNA viruses, the central dogma is important, is the rule, right? So that's our one rule. Of course, DNA to RNA, RNA to protein happens in 2 steps. DNA to RNA is called transcription, RNA synthesis. Remember, the word synthesis means to make. We're going to see this word. We've already seen this word a million times, right? And then RNA to protein is called translation or protein synthesis. And, you know, these words seem quite similar, not synthesis, transcription and translation. Sometimes it's easy to mix them up. Right? If you're not thinking, and when I'm on a tired day, I'll just say the first word that comes out of my head, and then I realize I said the wrong word, right? So, you know, let's define these words as what they mean. Transcription, or to transcribe, means to copy, right? Think of what DNA is. DNA is a nucleic acid. RNA is a nucleic acid, right? So really all we're doing is we're copying nucleic acids, right? So we're taking nucleic acid, and then we're making nucleic acid. Translation, as we know, is like changing languages, right? So now we're taking nucleic acid and we have amino acid or proteins, right? Totally different molecule, right? So translation, now we're changing the molecular language from nucleic acid to amino acid, right? And then so collectively we refer to this as gene expression. When we go from DNA, to protein with that RNA intermediate. Okay, and then of course, as we talked about, gene expression is extremely dependent on the environment, right? Some not so much, but many genes are not turned on unless we are under the quote-unquote right environment. All right, so we will talk about that, but first let's talk about replication, because we did talk about how, you know, in order for DNA to be copied, it needs to be replicated, okay? So here is the replication fork, and look at this. This is like nuts, right? We got these primases and polymerases and single-stranded binding proteins and Okazaki fragments, lagging and leading strands, and what the heck, right? So let's forget what we just saw. Okay, we will revisit this picture. We will come back and talk about some of the important elements of that picture, right? But we want to keep it simple, just enough to understand how replication works, just to understand the implications of replication in some other processes, right? We're going to keep it surface. So let's start off by reviewing DNA structure, okay? So DNA, we know double-stranded, right? We know it's a polymer of nucleotides. What are the 4? That's right, 4 nucleotides in DNA. Who's got the answer for that? Well, if you said adenine, thymine, cytosine, and guanine, you are correct, or A, T, C's, and G's. We talked about complementary base pairing. What is our complementary base pair rule, right? What is that? That's right, at the DNA, A to T, C to G. All right, that's pretty much most of what we need to know, at least for us. And then finally, DNA has direction. This one is a little bit more tricky, right? Talk about the 3' and 5', 3' hydroxyl, 5' phosphates, right? So here we have summarized more or less everything we need to know about DNA, right? The structure, complementary base pair rule, 3', 5', and then when we look at the structure, reviewing from our previous chapter, once again, you know, we like to look at DNA. This is how DNA, the cool picture with the the helix, right? But it's easy to think of it this way: A to T, C to G, right? And then remember, if one goes 3' 5' this way, the other goes 5' 3' this way, or we have direction, and then it's our 3' hydroxyl. That's super important because this is where we're going to add DNA to, always. Okay, so that's our super duper quick review, which we all remembered, right? So let's talk about our replication. And we're going to start off super simple. This idea of the parental and daughter molecules. Now, your book goes into this really silly deduction of semi-conservative replication, right? And it goes through like paragraphs, and at the end it's like, aha, it's semi-conservative, right? So let's just skip to the good part, right? Semi-conservative replication, right? And so we need to define our parental DNA molecule. That's what we start with. And we're going to make two identical daughter molecules, right? So the two new molecules are called daughter molecules. Now, we cannot just make DNA, right? It's what called de novo. We can't just make new DNA out of nothing, okay? So we actually need to use our parental DNA as a template. So it's going to act as the model for making our new DNA, right? And we do that using complementary base pair rule. If we have one DNA template, we simply are going to take the complementary basis to make our new DNA. All right, so then this lends itself to this. We have our parental DNA molecule. We're going to split that into our two strands. And then each strand is going to be used as a template to make our new daughter strands, right? And then so our new daughter molecule is actually half parental, half daughter. Semi-conservative, okay? And the rules, the way we fill these in are complementary base pairs. Okay, so let's talk about replication then. Couple more things to add, right? Replication has to start somewhere, right? So replication starts at what we call the origin of replication, or what we call the ORI, if you want to say origin of replication, too many words involved, right? And then our circular DNA molecule here, right? Notice this color scheme, dark purple. Here's our origin. Our replication is actually going to proceed in two directions, right? Around until we meet at the end, right? And then now we end up having two molecules. Notice each one is half dark purple, half light purple, semi-conservative, or dark purple parental strand or light purple daughter strand, right? Two DNA molecules, which then can be sorted into two cells, okay? Three basic stages. We have the initiation, which is the start. We have the elongation, where we grow our nucleotide strands, and then termination, where replication ends, okay? Initiation, elongation, termination, okay? The processes of initiation and termination are quite complex, actually elongation as well, but we're going to keep it super simple. It starts at the origin. We're going to add nucleotides, A to T, C to G, and then it's going to end, and at the end we have two daughter molecules, each of which is half parent, half daughter. And that's it, right? Kind of owing to its complexity, there are tons and tons and tons of components involved. I don't want you to know any of these except this one here. DNA polymerase 3. If you just put DNA polymerase, I will accept that as well. Okay, but it is DNA polymerase 3, or for all the cool cats out there, we call it DNA Pol 3. That's the main enzyme that adds nucleotides to our growing DNA chain. There are other DNA polymerases, of course, as the number implies, but we'll just keep it simple as DNA polymerase. So I will presume you're talking about DNA Pol 3 or DNA Pol 3, and then helicase. And what helicase does, this is the other one I want you to know, only because it's going to be a hilarious joke, as we'll talk in a second. Opens or unwinds or unzips the DNA helix, right? So our double strand, it takes our double strand and breaks it open or unzips it into our single strands, right? So if you're ever trying to flirt with another scientist, you say, oh, if I were an enzyme, I wish I were helicase, because then I would unzip your genes. And that's the only reason I want you to know the helicase, just so you can make and understand that joke. So let's revisit our replication fork. I know if my wife and son were here, they would be booing me right now. So if this is what let's revisit the replication fork. Look at all this complexity. Once again, ignore it. I just want to point out some of the important elements that we just talked about, highlighted by our text boxes. Okay. So first of all, this is kind of the overview. Remember, we're going to have replication going in both ways after initiation at our origin. Now we're at the elongation part. We're going to have it in both ways. Notice 2 strands of DNA. Therefore, we need 2 DNA pol 3s, right? So it's DNA polymerase 3 that's going to add nucleotides. Bang, 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 going this way. Bang, 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 going that way, right? So it's going to add nucleotides. And what it actually does, I drew those things in the wrong direction. Basically, I'm drawing the nucleotides coming in, but it's going to form the covalent bonds between nucleotides, right? Because remember, these are hydrogen bonds. Okay, so it's going to join up the nucleotides to form our new daughter molecules. Okay. The other thing I want us to know is DNA is added in the 3', added to the 3'. So over here we have our daughter strand, 5', and then the 3' end. We're always going to add it to our 3' end. Always. 3' end. Okay? So our new DNA then is read 5' to 3'. It's going to be made in that 5' to 3' direction. Always to the 3' end. Okay? Which then has this weird effect because remember, DNA is anti-parallel. One strand actually gets made, in this case, right to left. And our bottom strand here, in this case, gets made left to right. All right, so we have what's called the leading strand, lagging strand. I'm only saying that because I want you to just put that in the way back of your mind somewhere. So one day someone says, oh, we got the leading strand and the lagging strand. I want you to say, oh, yeah, I remember something about that. I have absolutely no idea what that means, but I do remember something about that. And we're going to just leave it at that, okay? And then finally, and this will be relevant a little bit later, we do have proofreading functions, right? So many of the other polymerases that are involved in proofreading, right? Because DNA polymerase adds 1,000 nucleotides per second, right? So 6,000 words per minute, right? So that's how fast DNA polymerase can add nucleotides. Actually, it's, I guess, 6,000 letters. Yeah, I know. I'm like totally hyping up DNA polymerase. 6,000 words a minute. Oh no, 60,000 per second. That's 60,000 words a minute. Actually, 60,000 letters, right? So, but it's still pretty fast, right? So it makes errors occasionally, right? But it can go back and fix some of the errors that it makes, right? And so we'll talk about proofreading a little bit later and then its implication, right, in our copying of DNA. And that's it. That's all I want us to know. Okay. Replication in prokaryotic cells happens pretty much the same. Initiation, elongation, termination, right? With a couple of differences. Remember, linear molecule, right? So we have linear molecules. And because the molecules tend to be much larger, we typically have more than one origin of replication. So we'll have our origins here, right? And then we have replication going out in each direction until they meet, right? And at the end of the day, we'll have these, once again, 2 parental DNA molecules, right? One, each of which are half parent, half daughter. Okay. The other interesting thing is just because of the way that DNA replication happens. Remember, it doesn't happen de novo, right? And it's this weird phenomenon to do with the leading and lagging strand. Hey, remember I mentioned leading and lagging strands, right? But you have no idea how it actually works. Good enough for now, right? There's this weird thing that happens where we get this overhang, right? And then we have enzymes called telomerases, which help fill in these overhangs, keeping our chromosomes the same length. But sometimes what happens is rather than adding in those extra nucleotides, we lose those extra nucleotides, right? So actually this is interesting. The only reason I mention this is because there's this interesting hypothesis that suggests that aging really is a product of the shortening of our chromosomes. And so as our chromosomes get shorter and shorter and shorter, our cell detects like, hey, these telomeres are shrinking. So maybe we shouldn't make as many of these proteins anymore, or maybe So some of these cells, these telomeres are way too short. Let's kill these cells off now because we have these super duper short chromosomes, right? And so we end up losing some cells because of the fact that the telomeres start to shrink, right? And so there are some interesting hypotheses as to increasing our telomerase activity, or figuring out a way that not only do we maintain our chromosome length, but how do we add to our chromosome lengths as anti-aging therapies, right? So I'm sure there are some science fiction works out there where in some science world, super rich people have figured out how to like double their chromosome length, and now they look like babies when they're 92, right? But anyways, I just wanted to mention that as point of interest, okay? Okay, so we're about halfway in. We talked about genetics and genomes and stuff like that. We talked about how DNA is copied. Now is a good time to take a break. Kind of let that sink in. All right, in fact, I'm just going to leave you with, let that sink in. Maybe go back and read the book a little bit, right? And then help fill in some of these gaps. Because next, what we're going to do is we're going to move on to our flow of genetic information. Okay, and in fact, my computer keeps crashing on me, so I'm going to take a break too. I'm going to make sure I save this video, and then I'm going to start a video again. But you, hopefully, will not notice. All right, so let's now go to our dogma of DNA. What does it even mean, right? So remember 2 steps, right? DNA to RNA, RNA to protein, right? What is the DNA to RNA step called? Anyone remember? Way back, either 15 minutes ago, if you didn't take a break, or if you did take a break, who knows how long ago? Transcription, yay, right? So transcription is the synthesis of RNA. And then once again, we don't make RNA de novo. We use DNA as our template. A couple things, RNA is single-stranded, uses U's instead of T's. All right, so that's important. And then what RNA does is acts as a copy of the information in our DNA, right? So what does that even mean, really, right? So let's think about what DNA is, right? It's that information, right? And the analogy we always use is a blueprint, right? And if you don't really know what a blueprint does, right? Blueprints are, the information to make everything. So you have a blueprint for your house, right? And this information for your house has, how many 2x4s do you need, to put up your walls, and where does your wiring go, where does your plumbing go, and all these things. You have like all these important pieces of information to make this house that you have, right? And so you have this architect who sits there and draws this blueprint and makes you your lovely dream house, right? Now, on site, you're not going to walk around with this DNA. Your blueprint, right? Because what if you spill your coffee on it, right? Or, the pipe bursts and water gets all over it and stuff like that, right? All of a sudden now you lost your one copy. So you actually have your master blueprint and you make these copies, right? And then it's these copies that you bring to the work site, okay? These copies are RNA. They're still pretty much the same thing. They're still paper. Typically they're white instead of blue. Yeah, they're called blueprints, by the way, because they're blue, believe it or not. So they're white, right, these copies. And then if you wreck one of these things, you know what you can do? Just go back to your original blueprint, make another copy, and then you got yourself a copy. And now this way, everyone gets one. The electrician gets one, the plumber, the concrete person, what are they called, masons. Everyone gets a copy of this blueprint, right? And you can always make more. So that's kind of the function of RNA. When we make RNA, it's complementary to DNA, right? And then remember this, U instead of T. So if you're ever looking at a piece of nucleotide and you see something like that, you're like, oh, I know that's DNA, bang, right? Because of the T there. Whereas, something very similar, right? Hey, I know that's RNA because of the U's, right? So that's very important. The other thing I want to mention is by convention, we always write our nucleotides in the 5' to 3'. So for us, that's considered the forward direction, right? Left to right if you're reading in a Western world. So we always write it 5' to 3'. And then we almost always only write one strand. So even with DNA, we know it's double-stranded, we will almost always only write it single-stranded, right? Because Why even bother writing that second strand? Because I know exactly what it looks like, right? What does my DNA strand look like? Why don't you do it right now? You can pause and fill it in. If you wrote TGGAGC, that's it right there, right? And that's our direction, 3', 5', yay. Good. The DNA that's used to make our RNA that serves as a template is called the antisense strand, right? Or the template strand. I prefer to use this word, the template strand, because it's the template strand instead of the antisense. For me, it doesn't make sense. So, you can almost say, hey, the template strand is also called the antisense strand, which doesn't make sense. And then the enzyme that you use is RNA polymerase. Okay, so it sounds like DNA polymerase. It polymerizes RNA instead of DNA. Similar to replication, has 3 steps. Guess what the 3 steps are? I'll give you 10 extra credit points if you can name it right now. That's right. Initiation, elongation, and termination. Oh, since I can't hear you, sorry, I can't give you those 10 extra credit points. 3 steps: initiation, elongation, termination. And just like with replication, initiation is the start, elongation is the continuing to make our long chain, and then termination is the end. Okay, so couple of things we want to add to that. So initiation in this case, remember for replication, it begins at the origin of replication. Here for transcription, it begins at the promoter sequence. And the promoter is simply DNA, right? So it's a sequence of DNA. So something like that, and people know what promoters look like, but like ATT. Oh, that's totally a promoter. That's our promoter sequence, right? So always understand that promoter is a sequence of DNA, right? And it kind of signifies the beginning of our gene. And then so we have RNA polymerase, almost always depicted as a gigantic egg. It's like, la, 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 la. Oh, look at that. There's a promoter. It sits right there. And then now it's going to go and move on to elongation. All right, so let's elongate something. All right, so a little bit neater this time, sorry. G, something like that. So here we have our template or antisense strand, right? And then complementary bases, just like before. Bases are going to come in. So let's fill in some of these gaps. Let's change colors. Just because I want to be like the cool kids. Ooh, I don't even know what cyan is. Let's find out. And then so now we are going to go and fill in our gaps. So T to A, oh pretty. A to T, right? Trick question. Not trick question. I tried to mislead you. U, remember, for RNA, G to C, C to G, G to C. And then RNA polymerase is simply going to go and form those phosphodiester bonds to our growing RNA strand, right? We're going to add that to our 5' to 3' direction. So we have 5'. We're always going to add it to the 3' end, right? And then that's going to go along until it hits ultimately, let's go back to red. All right, so because I'm running out of space. That's going to go until it hits our terminator sequence, right? So that's our terminator. And then so the polymerase is going to go and it hits this like, oh, that's our terminator. It's going to fall off. And then the RNA polymerase gets released. It's an enzyme. It can do this whole thing again, right? And then the finished RNA product gets released. That's what we call the transcript, right? So the RNA transcript is released, and then that's going to move on to our next step. Okay, nice and simple. Again, the regulation of initiation, the termination, the elongation, all these things, a lot more complex than we make it seem, but we'll just keep it nice and simple. Yeah? Good, so let's maybe take a picture that's much nicer than what I just drew. All right, so here we have our template strand or antisense strand. Notice our second strand does nothing for us, right? So we call this our non-template strand or our not template. Oh, by the way, I found an error in this diagram. So silly, which I will point out to you. Right, so here we have our promoter right over here, right? And then let's match these up, bang, A, right? So T to A to U, C to G, la 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 la, right? And it's going to keep on going along this way. And then C to G, A to U, T to A, G to C, those pauses are for you, by the way. T to A, all right, la 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 la, just like that, right? And then ultimately it's gonna hit, let's call this the terminator. Everything's gonna fall off, and then we have termination. Did you guys find the error yet? Oh no, wait, it's not an error because it's a weird and wonky. Why is it like this, right? Yeah, okay, here, look at that. How silly. I don't like how this is drawn, but that's not my problem. Okay, so super quickly then, if we're good with transcription, here we have our template or antisense strand. Let's make our RNA transcript. What does our RNA transcript look like? Pause it, fill it in. It should take you like not even 5 seconds, right? Okay, good. So it looks like G, C, T, A, T, T, C. Oh wait, what's that? Ah, I can hear you screaming through the screen. I tried to fool you again. You cannot be fooled, right? Use instead of T's, right? Good. Oh yeah, what happened there? All right, and then this of course will be 5' and 3', right? Had I drawn this way, what would our DNA strand look like? I hope you wrote it this way. C, U, U, A, U, C, G. Right? Because remember, we're going to add it. If this is 5', 3', remember, we always write 5', 3', right? So our new strand here will be 5, 3. So we're actually reading this backwards, right? So you always have to make sure that our finished product is written 5', 3'. So in this case, you're going to read it backwards. Okay, I know. Blows your mind. Okay, so now we've made our transcript. Now this RNA intermediate is going to be used to make our protein, right? What is the process of protein synthesis called? Those of you who said translation, correct now, because we're going to translate the language of nucleic acids into the language of proteins, right? mRNA here now, the RNA used to make proteins is called mRNA, and the M stands for messenger. Notice it's a little lowercase m, right? So we have different types of RNA, which we'll talk about very, very soon, right? The transcript used to make proteins is called messenger RNA or mRNA. When we now translate nucleic acids into proteins, we have to read it in words, right? And these words are called codons. Codons are groups of 3 nucleotides that we use to translate nucleic acids to amino acids, right? Basically, 3 nukes equals 1 amino acid. And we'll see how that works, right? And it's the sequence of codons on the RNA that then determines the sequence of amino acids, right? Which is our primary sequence, which then leads to our secondary sequence of folding, our tertiary as things start to come together. And then we have this completely formed, fully functional protein that now performs function, right? So at the end of translation, we'll have a protein or a polypeptide that goes on to do what it's supposed to do. And this conversion of codons to amino acids is what we call the genetic code. And in about 10 minutes, you will be able to read genetic code. That's right. I will teach you to read genetic code. Okay, so let's translate something. No, that's not true. Let's learn a few more things. Hold your horses before we can talk about translation. Okay, so in order to translate, we need a dictionary. Right, so this is our Rosetta Stone of sorts, right? Ingenious, by the way, how they figured this out, but that's for another story one day. Always written, our table is always written as mRNA because that's what we're reading. Always written in 5' direction, 5' to 3' direction, sorry. And in the way you want to read this is we have a first letter here, second letter, third letter. So if you have something like A, U, A, that's our first letter, A. Second letter U, right? So first letter, second letter, and then third letter A. There's AUA, and that's the amino acid that it codes for. That's a capital I, by the way, ILE. Okay? This table has 64 codons. 61 of these code for amino acids. So these are what we call sense codons, right? And so the way I like to think about it is it makes sense. These codons code for something meaningful, some sort of amino acid. Okay. Now, those of you who are clever will say to yourself, 61 codons, 20 amino acids. Hey, that means there are some amino acids that have more than one codon for it. And the answer is yes, very perceptive, right? In fact, almost every single amino acid has more than one codon for it, except for one. And I'll let you find out one amino acid that only has one codon for it. Some are hardcore. Look at this. Leucine has 6 codons that code for it. That's nuts, right? And so this is what we call the degeneracy of the genetic code. The fact that multiple codons can code for the same amino acid. So you can actually use and interchange different words, right? And still come up with the same protein. And this has a big implication a little bit later when we talk about what happens when we change our DNA, okay? The other 3 codons are what we call nonsense codons. Don't think of it as nonsense, as in like ridiculous. Think of them as not sense, nonsense. And these code for no amino acids. Basically, they code for stops. And sometimes we call these stop codons. Okay, so 61 codons coding for 20 amino acids, and then 3 stop codons. And when we get to the stop codon, guess what happens? Stops, right? All right, before we can start translating, we need to figure out or learn a little bit more about some of these components, right? So 2 major components are the ribosomes. And remember what the ribosomes do? They make proteins, right? This is way back in module 2. I'm going to say 2. Right? And ribosomes, they're kind of like enzymes, but technically not, because they're made up of both RNA, called ribosomal RNA. Notice the small r. Ribosomal RNA and proteins. So they're actually a combination of RNA and proteins, right? And there are multiple pieces to the ribosome, but there are 2 major subunits, the small and the large. And we call them that because the small one is small, the large one is large. Clever, right? Us biologists, like the things that we come up with. The other important component is something called a transfer RNA, right? tRNAs. And they're called tRNAs, clever, because look at that. It's like a cross, kind of like a T, right? But they also transfer things, and we'll talk about that in a second. 2 major parts of the tRNA we want to talk about. Way at the bottom over here is called the anticodon. And the anticodon is opposite of the codon, right? So if we have a GCC here, our anticodon here will be CGG, okay? And it's this part of that tRNA that interacts with our mRNA transcript, okay? At the other end over here, we have an amino acid, right? So we have an amino acid attached to this. So now notice we are speaking mRNA and amino acid, right? So this, the tRNA, in many ways acts as a translator, converting our language of nucleic acid to our language of amino acid. And let's figure out what this one is, right? So our codon here is GCC. GCC is alanine, right? So in this case, the amino acid that would be attached to that here would be alanine, right? So the tRNA brings the alanine to our ribosome, and then our ribosome is going to stick it together. Okay, so guess how many steps are involved in translation? If you said 3, congratulations, give your pat on the back. What are those 3 steps called, do you think? Nope, sorry. Yes, initiation, elongation, termination, right? So 3 steps. Once again, initiation, elongation, termination. Initiation is the start, elongation is the middle, termination is the end. Once again, like with transcription, a few important things here. Initiation starts at the very first AUG, right? AUG is a codon. It normally codes for an amino acid called methionine. But when we stick it at the beginning, it's something called formulated methionine, or sometimes F-met, right? As far as we're concerned, we can almost ignore that. Just think of it as methionine, okay? But basically, the very first codon that we're going to see, or that's going to start translation, is AUG, our start codon. Elongation then happens as we read in groups of 3. So let's build our hypothetical RNA molecule, right? We are going to initiate by finding our first. Oh, there's our first AUG. So this will now set our reading frame. This is where our first word is going to look like. And then after that, during elongation, what we're going to do is we're going to read these in groups of 3. And so on and so forth. Okay? And this is going to continue until termination when we hit our stop codon. At the stop codon, there's like this release factor. Everything falls apart. The ribosome falls apart. The protein falls off. We have just made a protein. Okay. So let's see again a much nicer picture, right? So here we have our AUG start codon. This will set our reading frame. We have our first tRNA carrying our first amino acid, methionine. Right, we have our large and small subunit. Notice how this one's large, this one's small. Yeah, and everything's going to come together here at our AUG. This now sets our reading frame, and so now we're going to read these in 3s, right? And then simply going to move along. A new tRNA is going to come in, bring amino acid. Another tRNA is going to come in, bring an amino acid, and then the ribosome is simply going to join our amino acids together until we get this fully completed polypeptide. So at termination, everything falls apart. Ribosome gets released, transcript gets released, and then our finally finished protein gets released. Okay, and we have made protein. Good. So how do we feel about this? Pretty good, I'm sure, right? So let's find out, right? Let's do a super I'm going to call it quick because I'm going to skip through a lot of it. Activity on transcription translation. So here we have our template strand, our antisense strand of DNA. You are going to transcribe and translate this. Okay. So a couple rules to this. First of all, transcribe this to make our mRNA molecule, and then translate the mRNA to find what we call a hidden message. And if you do this correctly, you'll find a hidden message. And if you did it right, you will know you did it right. If you did it wrong, its message will not make sense. Couple rules, right? When you add our amino acids, right, the first letter of the amino acid will spell out the message. And remember, normally we would start at our AUG, right? But for this, start at the beginning, because I didn't want to limit myself to a message that started with M. Okay, so pause it, and then I will solve it for you, right? Or if you're lazy, you can just watch me solve it, but just solve it for yourself. It's not very hard. All right, so here's our template strand. We are going to make mRNA. Remember, complementary base pair rule, A to U, G to C, right? So we have U, C, U, C, C, U. And then now I'm going to speed this up, and the next thing you're going to see is the fully formed RNA transcript. Okay, I made one oopsie, but that's okay. Hopefully I did this right. Sometimes, you just stare at these letters all day long. I used to do this in grad school. So easy to make a mistake, right? But hopefully we did this right. So that's what your mRNA transcript should look like. And then let's change colors. Let's go with magenta this time. Right, and then now what we're going to do is, remember, normally what we would do is like, la, 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 la. Oh, there's our first AUG. This would set our reading frame, right? But because of this exercise, we are going to start from our first one. We're going to find UCU. UCU's right there. Serine, so we have an S. CCU. CCU is a proline. That's a P. Okay, and I'm going to go through this once again. Next thing you will see is our finished polypeptide. Okay, we hit our first stop codon, UGA. Sometimes I'll see this. Don't write stop. It just stops. Sometimes I'll see this. Well, we got to stop, and then, oh, there's AGA, right? So I'm going to continue and put this in there, right? Nope, don't do that either. Remember, stop means stop. So if you did this correct, look at our spam is tasty. It totally is, right? I know I spelled tasty wrong, but limited alphabet, what can I do, right? Okay, so if you did it correctly, this is what you should see. If you did this okay, congratulations. You can now read genetic code. You can add that to your resume of languages that you know. I know genetic code. I just want to add one little thing. We call this an FYI, right? It's not necessarily going to be on the exam. Okay, by not necessarily, I mean it's not going to be on the exam. But just want to point out some of the differences between like reality of what we talk about versus reality of what actually happens, right? And then some of the differences between eukaryotic and prokaryotic transcription translation, right? Bacteria, for them, the ability to make proteins is incredibly important, right? So they jump on this thing like crazy, right? So in fact, they're so greedy that as they are making their transcript, ribosomes jump on there and immediately start making proteins. So this poor RNA polymerase is working to make this transcript, right? And then all these ribosomes already start jumping on it. It's like, hey man, I'm not even finished yet, but it's just jumping on there. And it's not even like one ribosome. Notice like as soon as another ribosome can squeeze in there, bang, another one comes in there, right? So there are all these little greedy little things immediately making protein, right? But it's super important for bacteria because they need to make proteins quickly, right? The speed at which they can respond to environmental factors is literally life and death for bacteria, right? So they're extremely efficient in how they do this. For eukaryotic cells, a little bit less important in the speed at which they do things. Very important that things get made properly and to the right place. Okay, so one of these things is the fact that, remember, DNA is in the nucleus, so we actually transcribe in the nucleus, and then that comes out to the cytoplasm where we make protein, right? So one of the things that needs to happen is that we want to make sure that when our transcript leaves the nucleus, it's the correct transcript, right? So there's something called RNA processing, where we actually make like this secret RNA, right, that it's not quite finished yet, and then so called the pre-mRNA, and then we got to chop that up a little bit, and then when it's finished correctly, it can leave, right? So it's almost like, oh shoot, you made a mistake when you made this RNA. Yep, I can tell you made this mistake because the pre-mRNA didn't quite look right, so it actually doesn't get to leave, right? The other thing that's important is that when translation happens, a lot of times, because you make a protein in one part of the cell, but you need to send it to a different part of the cell, we have to tag it with something. Or sometimes we want to stick it on the membrane, or sometimes we want to stick it out of the cell to another cell somewhere else. So we got to add these little tags, right? We talked about that with respect to the endoplasmic reticulum, the Golgi apparatus, all these little things. So proteins are also modified post-translationally. Okay, so a lot of these weird things happen, and sometimes you'll hear reference to glycosylating or adding lipids or all these various things, right? Post-translational modification, very important, right? But not on the exam. So you can choose to forget it if you want to, right? But I want to be the person that at least told you about it, and then you can choose to remember it if you want to or not. All right, so we have learned how to copy DNA. We have learned how to express a gene. Now we will talk about what happens to changes to our DNA. How does our DNA change? Okay, so once again, good time to take a break. Once again, I'm going to learn from my previous lesson. I'm going to take a break and save what I just did. And then I will either see you in about like 1 second, or I will see you in whenever you come back. All right, we're back, or we haven't gone anywhere, whatever the case may be, right? So let's talk about changes to genotypes, right? And changes to genotypes typically come in the form of what we call mutations, right? Now, mutations are, we often use them, and by we, I mean non-science people, but even sometimes scientists, this is irritating, use them incorrectly, okay? Mutations, by definition, are a heritable change in DNA. Okay, heritable is a weird word. Inheritable, right? It has to be able to be passed on for it to be a mutation, right? Sometimes DNA changes, and it's in some cell that never divides, right? There's some cell that will never ever persist. The DNA will never persist. Not really important to us, right? And it has to be changes in DNA. Sometimes we make errors in transcription. Doesn't matter. Transcripts, remember, temporary entities. Sometimes we make an error in translation. It's okay. Proteins, temporary entities. And we have so many proteins and so many copies of the RNA that if we lose, or if a couple of them are mistakes, no big deal. We have others that'll take care of it, right? But the DNA is permanent, right? So only changes in our DNA are considered mutations. Right? If an organism has a mutation, we call it a mutant. It has a change in genotype. And depending on where this mutation is, it may have a change in phenotype. Okay, so typically we only talk about mutants when they do have changes in phenotypes. But, really anything with a change in DNA is mutant, right? Of course, with that, then we have to refer back to what the original looks like. So we call the original the wild type, the most common, or the original phenotype. Sometimes a mutation is so beneficial that everyone has it. It becomes kind of like the new wild type. Okay, but regardless, wild type is what it used to look like. The mutant is what it now looks like. Okay, and there are several ways that mutations, several types of mutations, right? Which technically is two types, right? One is called a point mutation, and that's when we take one base and we change it with another, right? So sometimes as you're copying things, you're like A, C, C, G, and you get tired, like, oh, blah, right? And then you copy this thing again, you're like A, C, 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 A, and then whoops, I accidentally changed the G to a C, right? That will be a point mutation. An insertion is if you copy this and you completely forgot that G, right? Shoot. Right, so that's did I say insertion? I meant deletion is when we remove that base, right? The insertion would be I accidentally copied one extra C in there, right? That will be an insertion. Generally, changes to phenotypes are harmful. Some can be beneficial, right? So it all really then determines, or is determined by what is the effect of that mutation? What is the outcome? Okay, so let's look at each of these mutations and then see what the outcome is on a protein. So when we're looking at this figure, this is from your book. Here we have our wild type DNA, 5' to 3'. Super quick question. Is this the template strand or the non-template strand? Here we have the mRNA for this from this wild type DNA. So I'll let you figure that out. Right? Maybe I'll answer it later. And then here's the amino acid sequence for this super short 4 amino acid protein. Okay? So this is what it looks like normally. Sometimes what can happen is we can make a mistake in copying. So notice this A has now become a T. Okay? Which means then our resulting codon is GUA, which now has become GUU. However, luckily, when we look at the codon table, the protein or the amino acid that is coded by GUA is valine. The amino acid that's coded by GUU is also valine, right? That whole idea of the degeneracy of the genetic code, right? We can actually make these small little mistakes, and then it's not a big deal. We're okay. We're good, right? No harm, no foul. All right. So in this case, we would call this a silent mutation. There's a mutation, but no one really knows because we don't see it. All right. Okay. Sometimes, so in this case, we have another stretch of amino acids here, right? So we have, where are you? Oh, there it is. We have this C stretch here, but accidentally it got turned into an A, right? Which then has turned our CCC codon into an ACC codon. CCC codes for proline, ACC codes for threonine. Now what we have is called an amino acid substitution, right? Or a missense mutation. right? And missense in this case, remember sense meaning codes for amino acids, right? And missense is, oops, we coded for the wrong amino acid. Okay, so where now we used to have a proline, now we have a threonine. In this case now, these are the most interesting ones, right? Because what does this proline do? If the proline is in the active site of an enzyme and is very important in grabbing a substrate, maybe now by changing it to a threonine, we have lost that ability to grab that substrate. So we have an enzyme that doesn't even work anymore, right? Or maybe now, rather than being able to grab lactose as a sugar, all of a sudden now we've added the ability to eat, let's call it galactose, right? Or some other sugar, cellulose, right? So we've now gained this ability, right? So, sometimes they can be good, sometimes they can be bad. And it ultimately depends on the role that this amino acid does, and then what effect will have this new amino acid will have on the protein. Okay. Another thing that can happen is here we have a TAC, and this C gets changed to a G, all right? Changing our UAC into a UAG. And in this case now, what used to be a tyrosine now becomes a stop codon, a nonsense codon, right? And so that's what we call these nonsense mutations. And nonsense mutations are almost always bad, right? In this case, it's the very last amino acid that gets lost. So sometimes if it happens right at the end, sometimes we can get away with it. But generally, our protein becomes non-functional. So whatever role that protein played is not going to be useful anymore, right? What else did I want to say? Yeah, anyway, so non-functional proteins, right? And remember, changes to the DNA, right? Because these things now persist. The other type of effect of a mutation is called a frame shift. And these happen because of insertions and deletions, right? So now, let's see, here we have this TA, and then just to do some weird rando error, those TAs got removed, right? So what happens now is our whole reading frame shifts, right? So it used to be AGC, oops, let's go AGC, GUA, right? But we lost that. And remember, it doesn't go G, skip, and then CCC, right? This now goes GCC, and then this goes CUA. So we're changing every single amino acid after the deletion in this case. And of course, the same would happen is if we have an insertion, okay? So every single amino acid gets changed, right? So we're always gonna make proteins that are non-functional, right? Because somewhere along the way, we're either gonna run past our stop codon, right? Or we are gonna introduce a random stop codon. So we're gonna have this every single amino acid gets changed. We're gonna have this really weird looking protein, right? So mass destruction. So insertion deletion mutations tend to be extremely bad, okay? Okay, so these are the types of mutations, right? Point mutations and insertion deletions. That kind of then leads to, how does that happen? Like where do they come from, right? And so it turns out that most mutations are spontaneous, right? Most mutations are errors that occur during DNA replication. Now, DNA polymerase, remember, it goes real fast, 1,000 bases a second, right? And then with its error, and so it's bound to make mistakes because it goes super duper fast, right? But with its error correction, its ability to proofread, the proofread function, it actually makes very little mistakes, right? Only about one base in a billion copied, right? Now, well, think about this though. A human genome is billions big, right? So no matter what, every single time you copy your genome, you're gonna have some errors, right? Even though the error rate is extremely small. So you're gonna have introduce some errors, right? But the cool thing is that, remember, most of our DNA is junk. Statistically, a lot of the mutations that do occur will happen in areas that do not encode for genes, right? In fact, this is one of the many ways that we can use to use for DNA fingerprint analysis, or to identify you as the daddy. You're the daddy because I know this piece of DNA is unique to you, because as you've copied this DNA, and as you've kind of inherited these various what we call single nucleotide polymorphisms or SNPs, right? We can actually use these then to identify people, right? Because of the fact that we have all these little differences. Most of them are non-coding ones because generally any detrimental DNA or mutation that occurs in a coding sequence will almost always lead to something non-functional, which will almost always lead to a, let's call it non-living organism, right? Mutations, however, can also be induced, right? Sometimes things can happen in the environment that can increase the mutation rate, and these are what we call mutagens. Right? So mutagens are basically things that increase the rate of mutation, and so there are two general mutagens we will talk about: chemical mutagens and radiation, and then their mechanism really depends on what it is that they do exactly. Okay, so we'll start off by talking about chemical mutagens. And chemical mutagens typically interact with DNA somehow. Right? Somehow they're hanging out with DNA. Right? And there are three classes of chemical mutagens. One, the nucleoside analogs, base modifying, and intercalating agents. Okay, nucleoside analogs, nucleosides are like nucleotides. It's like, a couple things missing. Doesn't have the phosphate group, all that kind of stuff, but that's okay. We'll just kind of stick it as looks like a nucleotide. However, so it gets incorporated into a growing DNA strand. However, because of its slight differences, it mismatches differently, right? So here we have something called 2-aminopurine, and this is adenine. Look how similar, of course this is going to be the same. Look how similar 2-aminopurine looks to adenine. Almost identical, except for that N right there, right? So then this means that even though it gets incorporated like an adenine, it actually base pairs with cytosine. Instead of, what does adenine normally base pair with? Yeah, normally thymine, right? So normally what we'll have is this AT base pair, but then we'll have this AC base pair, right? Which then eventually we get turned into a GC base pair. So we've gone from this A to a G. Okay? Base modifying chemicals modify bases. So they react with bases and change some of their structures, right? So nitrous acid, for example, takes cytosine and turns that into a uracil, right? And does that by changing some of these, this NH2 into that O right there, which once again now will cause it to base pair off funny, right? Because here we have cytosine that's normally G, right? And then now this becomes a U, so all of a sudden now we have a UG, which is weird because it doesn't like it, both because it's RNA and because it's DNA. Oops, I didn't write that right. So that's U, that becomes a U, so now UA, I want to try to keep the same format there, right? And which then now becomes, after another round of replication, TA. So now we've gone from the CG to a TA, the C to a T. Okay, so once again, nucleoside analogs and base modifying chemicals will introduce point mutations because we are affecting one nucleotide. Okay, and then that will then lead to whatever, depending on which nucleotide we goofed around with. Okay. Intercalating agents insert themselves into the double helix. Right? So the way I like to always think of this as, in practice is, we have all hung out on a couch. There are 3 of you on a couch that clearly only sits 3 people. Yet we all have that irritating friend that just wants to sit with you guys. And so they throw themselves in between everybody, and then you're all squished in there, right? And it's like, you're so irritating, right? So what ends up happening? Well, sometimes you can fix it. Sometimes you're like, one person leaves. Right? But generally what happens is like everyone gets irritated, so at least 2 leave, right? So that leads to a deletion because, as you're trying to deal with this irritating friend that kind of mobbed the couch, you end up losing an extra person. So now you used to have 3 people on that couch, now you have 2. And then even in worst cases, someone else sees that and thinks it's hilarious, and they jump on the couch too, and all of a sudden now you have way too many. So now you have this insertion, right? So intercalating agents, in order to, as you try to fix the situation, can often lead to either deletions or insertions, right? Which then leads to frame shifts, right? So intercalating agents lead to frameshift mutations. Good? All right. Now, how does radiation work? Well, radiation really works by just goofing with DNA, like just destroying DNA, right? We talked about ionizing and non-ionizing radiation. Remember, ionizing, deeply penetrating, non-ionizing, not so much, right? And the way they cause mutations, slightly different, right? Ionizing just destroys DNA, causes these breaks, whether they're single breaks, double breaks, just all over the place, right? Just causes these major breaks. And of course, not of course. In fact, that is how gamma rays or ionizing radiation kills bacteria or you. It just like bombards you with so much radiation, causes so many breaks in DNA that just like, oh, well, this DNA is destroyed, right? Cell can't divide, cell is dead, right? But it's these sublethal doses that causes these small breaks, and we have mechanisms that'll fix it. It's like, oh, we got a little break, we can fix that, that's okay, right? But sometimes like, ooh, there's a little bit too much going on. What was here? I don't remember. And then you end up deleting a base, right? Or it's like, I'm pretty sure there was a T there too. So you end up adding a base, right? So, these types of damage can lead to either insertions or deletions. So once again, frameshift mutations. Non-ionizing radiation works a little differently. It has this weird effect of taking what we call adjacent thymines that are sitting next to each other, not across, right? So here we have bases across, and these two thymines are sitting next to each other, okay? And so UV light creates these covalent bonds between here, and then your polymerases, they don't like that. That's so weird, right? We do have mechanisms to fix it, once again, right? But if there's enough of these, look, these are also adjacent thymines, so if these things are also joined, right? Sometimes when you're trying to fix it, what ends up happening is that this gets lost, right? So once again, frameshift mutations, right? So radiation generally causes frameshift mutations as it induces either insertions or deletions. Okay, so we think of mutations as a bad thing, right? But actually mutations are vitally important because genetic diversity is the key to the survival of a species. If every single member of your species is genetically exactly the same, this species will never advance because one change in the environment that could be detrimental wipes everyone out, right? And I always think of it this way, as much as we want our friends to have the same interests and personalities, you do need some diversity because if everyone thinks the same, you will always eat the same food, do the same thing, talk about the same stuff, right? You need to be able to have some diversity. So for an organization to do well, you need diversity, right? For an organization to move forward, you need to have different schools of thoughts, different personalities, different everything. So when things change, your organization can adapt, right? So how does diversity get introduced? Well, it's through mutations, right? So the bulk of the diversity that we see in any species is through mutations, right? As these little things get introduced over millions and billions of years, for us millions of years, these small little changes happen, and sometimes they're bad and leads to, people that don't quite make it to adulthood. Sometimes they're great, and then they continue and persist, right? And then as we start to do what's called sexual reproduction, this genetic diversity gets mixed, gets created, and then we have this nice diversity, right? So for eukaryotic cells, you have mutations that create diversity, and then this sexual recombination that not only creates, that not only takes mixtures of two parents, but also whenever we make our gametes, we do a little mix before we make our gametes, right? Which is why every single sperm you make, every single egg you produce, they are all different, which is why every single child you have is different, right? Not just because you have DNA from the male and the female, also because every single sperm and egg is different from each other. Okay, so that's very powerful to create this nice genetic diversity. Prokaryotic cells don't do that. They don't reproduce sexually. They only reproduce asexually. So for them, mutations are really the predominant contributor to genetic diversity because technically all the offspring are genetically identical. And I do it identical in like quotations, right? Because mutations can change and really ensure the fact that your offspring are indeed slightly different. Because remember, there's always going to be differences, right? There is another way to introduce genetic diversity in prokaryotes, right? And that's called horizontal gene transfer, right? So here we have something called vertical gene transfer, where it's transmission of DNA from generation to generation. Horizontal gene transfer is transfer of genes between organisms of the same generation, okay? So it allows for rapid change in genotypes, much more so than mutations, okay? And some of these mechanisms are extremely powerful and a very important force, as we'll talk about in our next module. Most commonly, they occur between similar species, but they can occur between distantly related species. So now is a good time to take a break if you want to. Once again, I'm gonna save. I don't want to waste my work. I already lost 3 hours of work today. Stinks, right? But that's learning lessons. So I'm gonna take a break, and then I will see you in 1 second or whenever. So when we're talking horizontal gene transfer, we want to define a few things, right? We want to define the donor cell. The donor cell is going to give up its DNA. Right, so this is essentially the source of the DNA, and it's going to transfer part of its genome to our recipient cell. And so this is going to receive the DNA. Now, receiving DNA, it's in and of itself is not the final piece, right? You need to be able to take this DNA and incorporate it into your own genome, right? So think of it as like a house gift. Someone comes along and gives you this ugly lamp, right? You know, technically the lamp has entered your house, but if you take this ugly lamp and you throw it out, it is no longer part of your house, therefore not part of your genome, right? However, if you say, okay, well, it is for my mother-in-law, I'm too ashamed to throw it away, so I'm gonna leave it on the counter so she can see it every time she comes over, it is now officially part of your house, officially part of your genome, right? So that process we call recombination. And when you do that, the recipient cell becomes a recombinant. This one has changed genotype, okay? 3 ways this happens: transformation, transduction, and conjugation. We're going to bounce around the book a little bit for these because we've talked a little bit about some elements in different parts of the book. But let's start off with the simplest called transformation, okay? Your book, by the way, has no picture of transformation, or at least not a good one. So this one is from the old book. So if you are looking for this picture, you're not going to find it. Transformation is the uptake of what we call naked DNA. DNA that is not packaged, that is not wrapped, that is not enclosed in anything. It's just DNA. So here we have this recipient cell that's all like walking around, right, or crawling around in this primordial ooze, and then along the way it finds this DNA. It's just DNA, right? This recipient cell then takes up this DNA and puts it into its cell, right? Takes it up, but remember, this is like the lamp entering the house, right? Doesn't become part of its genome until it can recombine or become part of its own genome. And so when recombination occurs, now we have a recombinant cell. Okay, so now this recipient has become recombinant. We've changed genotype. And the change in phenotype would be dependent on what gene specifically it took up. Okay? So, really quickly again, recipient cell finds naked DNA, takes up naked DNA, recombines, becomes recombinant. That's transformation. One thing that seems to be missing is our donor cell, right? This is our donor DNA, and it says DNA fragments from donor cells. Where the heck is our donor cell, right? Well, it turns out that cells in general, they don't throw DNA out. DNA is precious to us, so we don't just throw it out, right? So in fact, DNA really only enters the environment after cells die, right? So after cell death, cells lysis occurs, and then the recipient can find this DNA and take it up, right? It's this ability called competence. Okay, so some bacteria have this ability to take up DNA. So in the wild, we have bacteria that are naturally competent, able to take up DNA. via transformation. So most recipient cells in the wild are naturally competent, right? But we can also do this in the lab artificially, something we call artificial competence. And one of these landmark experiments is the Griffith's experiment, right? This Griffith's experiment was the first experiment to show that there's some sort of thing that they call this transforming factor that can go from between bacteria and change what they look like. Okay, so I just like a lot of things, there's certain things I don't like about your book. They don't really define very well. That's not true. In the figure, they don't define what the S strain is, right? So the S strain, also called smooth strain, of this bacterium called Streptococcus pneumoniae, right? I.e. causes pneumonia, is pathogenic, or another word for that is virulent. Okay? There's a related strain called the R strain, right? Genetically slightly different, all right? Also called a rough strain, and I'm just going to write not. It's not virulent. Okay? Same bacterium, genetically slightly different. Phenotypes, one looks smooth, one looks rough. Okay? So that's kind of the setup for the experiment. So these are in mice models, right? So we have the rough strain. We can take the rough strain and we can inoculate or stick it into the mouse. Okay? Remember, that's our non-virulent. We can also take the smooth strain, which is virulent, right? We can heat kill it, basically boil it, and we can stick it in the mouse, right? So what do you think happens to the mouse that got stuck with the rough strain? Well, if you said, the rough strain is non-virulent, so therefore there's no disease, mouse should be fine. The answer is correct. Mouse lives, right? What do you think happens to the mouse that we stuck with the heat-killed smooth strain? Well, if you said to yourself, well, I mean, the smooth strain you said was pathogenic or virulent. We killed it. Unless it's a zombie, nothing could happen. Mouse should live. And you're right. Mouse lives, right? So now let's get to the good part. All right. Griffiths took the rough strain. Remember, doesn't cause disease. Took the heat-killed smooth strain and stuck it into the mouse. All right. Now we all know something's going to happen, right? But what do you think that something is? Sorry for you sensitive folks, right? But this mouse got pneumonia and died. Oh, poor mouse. Right, and the cool thing is, not cool for the mouse, you can get tissue samples from the lungs, and then inside, they were able to recover live smooth strain bacteria, even though there were no living smooth strain bacteria inoculated or put into the mouse, right? And then, just to prove and complete the circle, they can take this smooth strain, stick it into a mouse, right? So this is the smooth strain that was now recovered from the dead mice. And then this mouse died. Okay, so they deduced that there's this transforming factor that went from the dead smooth strain to the live rough strain, and then it was able to transform it from the non-virulent strain to the virulent strain, right? The cool thing about this is well before we knew DNA was the genetic component. This is before we even knew what DNA looked like, right? Well before Watson and Crick, right? So it's kind of neat that they knew this stuff was going on, right? They all thought it was proteins because protein was kind of important, but later they realized, no, it's definitely not protein. It's gotta be something else, right? And so it wasn't until the structure of DNA was solved that they finally realized this is how it works. It's DNA. Okay, so that's transformation. Let's talk a little bit about transduction, right? We got to go back in time, way back to module 3, I believe it was, when we talked about viruses. So this is section 6.2 of your book. Transduction happens as DNA transfer that happens as part of a bacteriophage infection. Remember, bacteriophages are simply viruses that infect bacteria, right? Two types of transduction, one called generalized transduction, one called specialized transduction, and the only difference is that in generalized transduction, random segments of DNA are transferred, and specialized transduction, specific genes are transferred. I don't want to talk about how or why this happens, just that it does, right? So, for us, we'll keep it simple. It's the same thing, except random genes versus specific genes, right? And again, you can go and look at the lysogenic cycle in 6.2, which is this figure right here. I don't like this figure because the color scheme is terrible. So don't copy any of this. The exact same slide is right here. Okay, so I like this one much better. Transduction starts with a normal phage infection. Okay, so here we have a phage with its pink phage DNA. It's going to infect this donor cell. Here's our donor cell, just doing what it does. And remember, we talked about phage infection, right? It's going to spit its DNA into the cell. This DNA now is going to take over the cell make many, many copies of the phage heads, many, many copies of the DNA, and then put it all together to make many, many copies of the phage, ultimately bursting the cell and then releasing the phage into the wild, right? Every now and then, sometimes, the phage head gets packed with bacterial DNA. And this is what we call a transducing phage. This phage has bacterial DNA instead of phage DNA, right? So now, because it's the phage head that will drive infection, the phage DNA the transducing phage infects a recipient cell, injects its bacterial DNA. Luckily for the recipient cell, the bacterial DNA will not cause infection, will not cause it to die. In fact, if it can recombine this new bacterial DNA into its genome, it has now become recombinant. Right? So it's now a recombinant cell. Okay? Some things that are sometimes confusing to people. The phage is not the donor. It's this bacterium that is the donor of the DNA. The phage is merely the vehicle that carries DNA from the donor cell to the recipient cell. Okay? Normal part of the viral life cycle. The interesting thing about this is this actually happens on a eukaryotic level. In fact, with humans, right? When they sequence a human genome, they found evidence of many, many, many viral sequences embedded within our genome, right? And so the hypothesis is that we got infected by these viruses. These genomes kind of stuck in us. The virus never felt like leaving, and it just kind of stays within us and has now officially become part of our genome. So along the way, over the many, many years, we've accumulated these viral genomes, right, incorporated into our own, changing our genotypes. Okay? All right, so that's transduction. Now let's think back to transduction, right? Kind of an accidental process, right? Our donor got infected and then kind of accidentally packaged bacterial DNA in the phage, right? And so our recipient was lucky enough to get infected by transducing phage, so it doesn't die. I think about transformation. Our donor died, right? So in both of these cases, our donor, even though we think, oh, so nice, they donate DNA, right? They never intended to give the DNA to the recipient. Okay? The next mechanism of horizontal gene transfer, conjugation, is very different. It's a very active process, very purposeful. The donor wants to do this, right? In order to understand it, let's talk a little bit about plasmids first, right? I think we've mentioned plasmids. We're going to talk about just a teeny tiny little bit more. Right, plasmids are what we call self-replicating circular molecules of DNA. That's usually our definition of plasmids, right? They're very small, right? 1 to 5% of genome, sometimes even much, much smaller. And oftentimes they carry useful genes, right? And many plasmids are what we call conjugative plasmids. The genes for conjugation are found on the plasmid itself, right? And so the cool thing about plasmids is, first of all, when we say self-replicating, they copy themselves whenever they want, right? So while this bacterial chromosome is copying itself, these plasmids are like bang, 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 copying themselves so many times. So often we have hundreds or thousands of copies of a plasmid in any one cell, right? The other cool thing is, for it to be able to propagate and go from one cell to the next, it has all the genes that it needs. Right? In some many ways, some people like to look at these things as like a super rudimentary life form because it's DNA. It's able to be passed from one to the next and has genes essential for its own survival. All right, so really cool stuff. But we're not going to care about that so much. We're going to care about its role in conjugation. Okay, so conjugation is plasmid-dependent DNA transfer, right? And you know the way we always think about this, right? Because for biologists, it's always about sex. This is bacterial sex. Okay, we have cell-to-cell contact must happen, right? So before with transformation, with transduction, cells never see each other, right? Here, not only do the cells see each other, they must make contact, right? We have mating types, right? So our donor cell carries the plasmid. Our recipient cells do not carry the plasmid. So you cannot have mating between cells that don't carry plasmid, obviously because there's no plasmid involved. But you also can't have conjugation between cells carrying the same plasmid because the plasmid doesn't want that to happen. It wants to go to a cell that doesn't have the plasmid, right? And then check how this happens. Our donor cell seeks out our recipient cell, forms this conjugation pilus, right? And this pilus is simply this hollow tube that connects two cells, right? And then through that hollow tube, it injects its DNA from our donor to our recipient, right? I mean, holy crow, this is bacterial sex, right? I mean, how much more like sex can this be? Oftentimes the example used to illustrate conjugation is something called the F plasmid or the fertility factor, right? So even back then, before they really knew what was going on, they knew that somehow these cells were able to mate with each other, this idea of fertility. And then, so they call the mating types F plus, meaning it carries the plasmid, F minus, meaning it doesn't carry the plasmid. And it can be anything, right? You can have a plasmid called, very famous cloning plasmid, PUC19, which actually is not even conjugative, but that's okay, right? So if you have two cells, PUC19 plus, PUC19 minus, you can say this one here carries PUC19, this one does not, so then we can have conjugation go this way, okay? Conjugation is relatively straightforward. We have our donor cell, right? It's in the club looking for a recipient cell. It's like, hey, I'm helicase because I can unzip your genes, that kind of stuff, right? Recipient says, I like. And then so the donor cell will extend the pilus to the recipient, right? That hollow bridge. And then this is a very important part. We have something called rolling replication. The plasmid gets replicated as it gets transferred, right? So our recipient will have a single-stranded copy of this plasmid, right? And then once conjugation is finished, now it'll complete the second strand, right? And then we have an F plus cell here, an F minus cell here, and at the end of conjugation, we have two F plus cells. Okay, so now not only does the plasmid transfer, it actually makes its way into two different cells. So it's replicated itself as it gets transferred. Okay? Again, very active process, has mating types, donor seeks out recipient. So a donor is a cell that carries a plasmid, recipient does not carry the plasmid, and it's the genes for making this pilus for conjugation are all found on the plasmid itself. There are some special cases that I want to talk about, but not in detail. You can read it in your book if you want to. It's really cool stuff, super cool. Like everything. I love genetics. But sometimes this F plasmid can integrate into the chromosome. This happens, right? And when that happens, we call this HFR, high frequency of recombination, right? This HFR cell is still F plus, right? Because the genes for conjugation, remember, are on the plasmid right there. Okay. Now, when DNA can go into things, it can also go out of things. Excise is what we call it. Sometimes when that happens, the cut is a little bit imprecise, right? Because it didn't happen quite well, right? So you can actually have a case, and this is what we call an F-prime cell, right? Where we have the plasmid. It took a little bit too much chromosome in this case, oops, right? But once again, it can initiate conjugation, right? So both of these can conjugate, Hfr and F-prime, right? It happens slightly differently now because of the way that the plasmid is located. And so the genes that are received are slightly different, right? And I really don't want to take it much farther than that. But both Hfr and F-prime cells are still technically F+. They can still initiate conjugation. All right. So the very last slide, I promise you, this is the very last slide. All right. Plasmids are super impactful in so many different things, right? So plasmids themselves are kind of tedious to maintain. You know, you imagine a cell that has to constantly copy this piece of crap plasmid. It's like, holy, this crap plasmid came from nowhere. And I got to keep copying this darn thing, right? And copying DNA takes energy. So you got this stupid cell who's going to deal with this stupid plasmid and constantly got to copy it, right? It's going to use up all this energy, right? Whereas a cell that doesn't have a plasmid doesn't use that extra energy. So in this competition, the cell with the plasmid will tend to do worse, right? So plasmids then actually have, along the way, been selected for those that are carrying useful genes, right? So many plasmids can contain virulence genes. So for example, our plasmids, they're special plasmids for us because they encode genes that encode antibiotic resistance, right? So normally here you are like, hey, I can resist penicillin, and all your friends that don't have that plasmid are like, there's no penicillin around here. Stop being such a weirdo, right? But then all of a sudden, oh, here comes penicillin, and then they're all dying. You're like, hey, I was ready for it because I have this penicillin resistance, right? And so we'll talk about this more in our next module, right? Plasmids can be a very major driving force for increasing the antibiotic resistance in bacteria, which is a huge clinical problem. It's kind of a huge societal problem. Sometimes toxin genes can be found, right? So this idea of virulence, the ability to cause disease, right, can take a normally harmless bacterium like E. coli. We all have E. coli. Let's not forget that. E. coli is our friend, okay? But E. coli has these family members, let's just face it, they're not good people, right? Or they're not good bacteria. And these are the ones that have picked up little things along the way. And there's one strain called VTEC, is one that has acquired toxin genes from a related bacterium called Shigella, right? Shiga toxin, appropriately named, right? And it gets that through a plasmid. There are other types of E. coli that can get other virulence factors, again, via the acquisition of plasmids. Sometimes there are bacteria that can that have figured out how to eat really interesting carbon sources like crude oil, for example, or some sort of chemical lubricants. And these things can be on plasmids, and if they are, they can be shared amongst the populations. Like, hey, look at this cool thing that I found, right? And for me, the super coolest plasmid is this one by this bacterium called Agrobacterium. And it has figured out how to take this plasmid and transfer it to a plant. Not even other bacteria, to a plant, right? And so why would it do that? Well, it turns out that when it makes its way into a plant, it causes these tumors in the plant. So this agrobacterium hangs out like near the roots of these plants, right? And then it shoots its plasmid into the roots, and then all of a sudden these tumors form, and these tumors then have this tendency to split cells open, and when the cells split open, all these lovely sugar juices kind of fall out of the plant, and then there's agrobacterium on the outside like, hey, I get to eat it all, yummy, 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 right? And then eats all the lovely sugars from the tumors that it has induced, right? And this is actually a really neat mechanism for like doing a lot of molecular biology in plants, right? And then that leads me to the last one. Plasmids, vitally important in molecular biology. So many things, so many of our therapeutics that are developed, these vaccines that get made, all these new chemicals, new antibiotics that get made, all these things, insulin, all of these are dependent on using plasmids in the lab to be able to clone genes, to be able to do very useful things with it, right? So plasmids are this important, they're probably more important in molecular biology than they are in the wild. That's how important they are to us, even though they're super important to bacteria. Okay, so that's it for genetics, right? Genetics, one of my favorite topics, right? But again, try to keep things, like try to keep pictures in your mind. So don't read words and say, the plasmid gets transferred through this pilus. Like try to imagine, what do you think a pilus looks like? And it really doesn't matter if it's the right picture or the wrong picture. But you just want a picture that helps you kind of envision what is going on, right? And that helps you definitely keep track of some of these things that are hard to conceptualize and let you conceptualize it in a way that makes sense to you. Okay? So that's it for Module 7. I will see you very soon in Module 8, where we will talk all about this war against bacteria. We have ways that we can destroy bacteria because they're trying to destroy us. They have ways that they can fight our own chemical, or the chemicals that we use, right? So it's all about antibacterial chemicals, antimicrobial drugs, and then this warfare that we have with respect to these things. And some of the things we talked about in this chapter are going to be, or module, are going to be applicable to Module 8. So until then, good luck on your quiz. Have an awesome day.